Okay, another application of your PCR is for a process called RT-PCR, but in this case RT doesn't mean reverse transcription. It means real-time PCR, real-time. So real-time PCR, what does that mean? It's the actual measurement of PCR amplification during the PCR cycle. So by measuring how fast you can amplify out your PCR product, you can quantify amount of the template DNA that was present within your sample. Okay, so as a kind of quantitative PCR process that will allow you to measure the amount of template DNA that was present within your sample. That's what quantitative RT-PCR means. Okay, so again, you need to differentiate this process from the previous one, which shows RT-PCR. Whenever you say RT-PCR, that means reverse transcription PCR, which is used to measure the amount of gene expression happening within the living cell, right? That's what RT-PCR meant. But in this process, when you say real-time PCR, that's another type of PCR that's going to allow you to measure the amount of the template DNA that is present within your sample. Okay, so how does that work? So this RT-PCR is utilizing some fluorescent signal technology. So during your PCR process, there is involvement of fluorescent tag that is given, maybe added to your PCR product. Okay, or maybe it may be falling off of your PCR template DNA. So whichever it is, there are a couple of different ways of doing it. Whichever it is, this fluorescent signal molecule will allow you to measure the amount of the PCR product that is being made during your PCR cycle. Okay, so here is that how it works. So okay, so during your PCR cycle, okay, which is happening within all these wells, 96 well plates, the PCR is done in each well. Okay, so in each PCR you are adding some fluorescent tag in there. Okay, so by measuring the amount of fluorescent that is incorporated into your PCR product, okay, so you can see how much of the fluorescent tag is being added to your PCR product. Okay, so you can measure that during your PCR. At real time comes from that real-time measurement during your PCR. Okay, you can detect how fast you are making the PCR product. The rate of a PCR product, I guess, development. Okay, the speed is being measured. Okay, so that speed can be compared with maybe another tube that has another PCR setup. So the difference between this control PCR and maybe positive PCR or negative PCR would be negative PCR would not have the template DNA added, right? And the positive PCR may be the real sample. You are trying to measure the amount of the DNA, the template DNA present, and that may be the control. Control maybe is the PCR reaction where you know the amount of certain template DNA that's added, right? So that's the positive control, if you will, and that's negative control. So no template DNA added. Certain amount of template DNA added, and then in between you will do a measurement using the sample. In the sample you don't know how much of the template DNA is present, right? So you can compare the rate of the PCR product being made from maybe negative control, positive control, and from your sample. Okay, if your sample gives the fluorescent incorporation rate that's similar to the positive control, then what does that mean? By comparing that curve against this curve that came from the control, especially the slope, and the slope, okay, you can estimate how much of the template DNA was present within your sample. Okay, so it's a kind of quantitative way of measuring the amount of sample present using a PCR. So where do you use this method? So you're trying to detect amount of maybe DNA or RNA present within your sample. So for example, detection of pathogen like HIV virus, how much is it there in maybe maybe a given sample maybe came from a patient or something like that. Or in this case, right, so you're trying to see if there are some pathogens that are present in your food material, maybe hamburger sample taken from restaurants. You're trying to see if there's some food poisoning happening at restaurant businesses, right? And then you will compare those samples taken from this food maybe materials, and then against maybe negative control, positive control, and then you'll try to quantify how much of the PCR product you will make, just like this curve. okay? And then if the samples you have taken from the food material is giving you the curve like this, then you can compare the rate and quantify amount of DNA present. So most likely you'll be measuring possible, food pathogens like salmonella or E. coli or whatever from these samples. So it's used in quantifying amount of DNA present within your sample. Maybe it's a pathogen or whatever the problem that is caused. So that's the RT-PCR, real-time PCR. That's a quantitative analysis method. Okay. Another application here is genotyping. So think about the forensics, so where DNA fingerprinting is used, right? When you say DNA fingerprinting, what do you mean? So in that DNA fingerprinting analysis, you use a PCR process in there. So people's genomes are similar in the gene coding regions, as you know, but around the areas of non-gene coding regions, people do have unique DNA sequences. So we call those maybe variable nucleotide tandem repeat sequences, or short tandem repeat sequences, polymorphism, or single nucleotide polymorphism. You find those unique DNA sequence bits from individual genomes. Okay, so that's those are the side of our genomes where those DNA fingerprinting is targeting. Okay, when you compare my DNA against any of your DNA, maybe the gene coding regions like exons, their sequences are similar, probably about the same. They are very homologous, but non-gene coding areas like intron sequences or some other non-coding region within our genome, you will be able to find these segments where we'll have unique DNA sequences. Okay, so if you target your PCR primers around that area and compare the sizes of those areas or sequences of those areas, then you can find the unique profile of our DNA, and that uniqueness is so different. So the chance is only like 1 out of maybe billions people you will find a match. So that's how it is adapted in the crime cases in the the legal suit. Okay, so we use PCR for that. Okay, so STR regions, if you have primers amplifying on that area, depends on how many repeats you have. Maybe 5 repeats or 6 repeats versus 3 repeats. The PCR product size will be different, right? Same goes with this. Okay, no product versus product depends on the uniqueness of the sequence. This primer may bind or may not bind. So that's how you can use the PCR analysis in this DNA fingerprinting case. And then DNA PCR can be used in introducing mutations, especially point mutation. You're targeting one side of your gene sequence. You're going to change that. PCR is really handy. We talked about it in the beginning as well, right? So yes, you can generate a primer with one nucleotide mismatch. Okay, top primer, bottom primer, and then have them anneal, and they will go around the opposite direction. So you will make the products, okay, and those products will be denatured, and then renature again. Then you can form this hybrid, and once you form the hybrid, you can fill those hybrid up by using DNA polymerase. So you can make the DNA double-strand sequences with one nucleotide mismatch. That's the mutated nucleotide set. If you clone that in, now you have the gene sequence with one DNA nucleotide mismatch. That's the site-directed mutation. You just use PCR for that. Okay. And then another way of disrupting one-step gene disruption. So gene disruption means it's a mutation process. Gene sequence will be mutated. That's what gene disruption means. You can use PCR process for that. In this case, instead of changing one nucleotide within your gene sequence, you can change a big segment of the gene sequence. You can either delete that, or you can insert a foreign gene sequence in the middle of it. That's what you mean by disruption technique. Okay, so you're adding maybe a foreign DNA piece in the middle of a gene coding region, then that insertion will cause a mutation. That's a gene disruption technique. So you can use a PCR for that. How does that work? So let's explain it by looking at the diagram. Okay, so if you have two primer sets, one primer set is binding on that sequence of the template DNA, and then within your template DNA you have your target gene sequence there. So one primer set is binding on that area, so it's going to amplify out that area, okay, and the other primer set, top and bottom primers, binding on these two sites, so it's going to amplify out that region. So you can amplify out this A segment and B segment from the template DNA through PCR, right? And then so you can assume that between the A and B segments, okay, so there is gene sequence, target gene sequence, so there's about that much of the kind of area that's empty between A and B, so it's that region, right? And to that region, that empty gap, you can fill that empty gap with some foreign DNA sequence. For example, this plasmid DNA, okay, can be cut using an enzyme like BIOT211 and E-qual1. So maybe those two enzymes will cut here and here, and you have the segment made. So if these two PCR products had the primers that will give you the BIOT211 cut site, E-qual1 cut site, now they will have what? The matching cohesive bands, right? Then you can easily insert this foreign DNA piece in the middle of it, so through the ligation process. So now A fragment, B fragment, in between there's a big fragment came from the foreign DNA of plasmid DNA piece. So now you have that all three pieces put together. So what is the importance of this? Somewhere in the A DNA there is a gene coding region. So with the starting with the ATG, okay, start codon, and then it goes on to that direction. And in the middle of it, there's a foreign DNA inserted, and then it continues. There's another part of the target gene. So in the middle of that, there's a foreign DNA just inserted. That's an insertion mutation. That's what it just created. That's a gene disruption technology. So where is the PCR used? So here, when you amplify out this fragment and that fragment, the PCR technology is used, and not only that, it gave you the nice enzyme coding sequences through your primer. So this primer has that extra sequence, that primer has the extra sequence for BIOT211 and the E-qual1. So the PCR is handy for that. And then finally, random mutagenesis. Okay, PCR can be used when you can randomly mutagenate your gene sequence. What do you mean by random mutagenesis? You don't know where you're going to target your mutation. You're going to change a lot of sequences within your gene randomly. Okay, so that's so-called random mutagenesis. Okay, so how is PCR handy for that? You create a PCR condition where the tag polymerase will make a bunch of mistakes. Okay, so it's called error-prone PCR. Okay, so create the condition where tag polymerase will make a bunch of mistakes during its replication. So examples are what you're going to add the nucleotide GATC in not equal ratio. You're going to add an unequal ratio of GATC. Some of them will be added a lot, some of them will be added less. Okay, then yeah, the tag, since there are some depletion of nucleotide, it's going to add whatever nucleotide is available. That's going to create a mistake. And then use high concentration of magnesium ions. That kind of inactivates your tag, and it's going to make a lot of mutations as well, mistakes as well. So these conditions will create more mutation through tag's mistake. So you can randomly create a bunch of mutations. So the result is that between those two primers, you're going to do a PCR, but now there are a lot of mistakes being made. So when you do that, PCR product will have a bunch of mutations in there. So that's how you can generate random mutagenesis using PCR. So that's another application of your PCR. Okay.